Here we are, Unit 3, Video 2, and in this video we are going to be looking at the environmental impacts on enzyme function and cellular energy, which is Units 3.3 and 3.4 of AP Biology. So let's talk about enzymes. Here we have a diagram of an enzyme acting normally, so you can see the substrate is fitting into the active site of the enzyme. But sometimes there are inhibitors of enzymes that interfere with the enzyme function. There are two types you need to know, competitive and non-competitive inhibitors. So what are those? A competitive inhibitor or competitive inhibition is where you have a molecule that interferes with the active site itself. So it's called competitive because then the inhibitor and the substrate are fighting over the same region of the enzyme, that same active site. Non-competitive inhibitors interact with the enzyme at an allosteric site, so somewhere else. That can change the shape of the enzyme such that the substrate no longer fits in the active site. But it's non-competitive in the sense that they're not fighting over the same location. And inhibitor molecules, I should point out, some can bind reversibly so that it can be, they can kind of come and go depending on the conditions. Sometimes it's an irreversible reaction. Next, let's talk about denaturation. So what is protein denaturation and what can cause a protein to denature? Remember that a protein's function very much depends on its structure. And so if you have a change in the protein's structure, that can interfere with its function. So denaturation, or a protein being denatured, is when you have loss of biological activity due to a change in the structure. This is usually a result of a change in pH or a change in temperature, and we'll look at those a little bit more closely in a bit. But first, I want to make sure that we understand how to analyze rate graphs. So let's take a look at an example. So before we talk about biological rate graphs, let's look at one that might be a little bit more familiar to you. So take a look at what this graph is showing, and also use this graph to help explain the relationship between temperature and the airplane's initial rate of climb. And finally, why would we want to choose to graph the initial climb rate? One of the things I hope you noticed about this graph is that there are three variables being shown. On the x-axis is minutes after takeoff, so this is time. That's one of our independent variables. On the right, you can also see the legend representing three different temperatures. So temperature is also an independent variable. And both of those are affecting our dependent variable of altitude. So what we're looking at here is how an airplane's altitude is affected by both time and temperature. Now, if we wanted to create a rate graph, we can do that by looking at the slope of each of these lines. But conceptually, you can actually feel this in an airplane because it's kind of how quickly you're climbing. It's the change in altitude divided by the change in minutes. So it's the rise over the run. So that's simply slope, but you can think about this conceptually by what it's feeling like if you're in that airplane. So you can notice at the beginning, all of these airplanes have a positive slope. So they are all gaining altitude as we go into the future, right, over time. Eventually, those are going to plateau. And that's why we want to look at the initial climb rate, is eventually things are going to plateau. So for a while, when we're at cruising altitude, there's not going to be a change in altitude. So if we're looking at how the temperature affects the rate of climb, we want to look at when there's actually a change in climb or a change in altitude. So we want to look at that initial climb rate. So now let's actually see the graph and see if this makes any sense. So if we look at the graph on the right, we can see the effective temperature on initial climb rate. And you'll notice that the optimal temperature has the highest climb rate, and we can measure that in meters per second. Too cold has a slightly lower climb rate, and too hot has a much slower climb rate. We can understand this conceptually if we look at those planes sort of towards the beginning of their takeoff, if they have very different slopes, right? So if you're in the plane that is too hot, it's rising very slowly. Whereas if your plane is too cold, it's rising pretty quickly, but still not as quickly as a plane that is at that optimal temperature. Right? Something else I should point out is that we've made temperature into a categorical variable. Temperature is usually a continuous variable because it's usually quantitative. We usually measure it with numbers. But in this case, we've made it categorical because we've just used qualitative categories of too hot, too cold, and optimal. And I wanted you to see this because we were just doing sort of a thought experiment. Usually when we're looking at temperature, we do measure quantitatively. So now we're looking at rate graphs biologically. Here we're looking at enzyme activity, or the rate of reaction of enzyme-mediated reactions, and looking at those rates. So again, this is like looking at the rate of climb of those airplanes. So notice that the x-axis is not time, but it's some environmental effect or some environmental factor that affects that rate. So let's see if we can get through all of these questions. Looking at the large graph in the middle of temperature, how does a low temperature affect the reaction? How does a high temperature affect the reaction? And then looking at the middle, what is the importance of that dashed line? And pay attention to that temperature axis when you're answering that one. Over on the right, you'll see pH affecting enzyme activity. And think about how is it that pH might have a role? Why would pH affect enzyme activity? Then why does enzyme activity plateau at high substrate concentrations? We can see that at the sort of graph on the lower right. And finally, not all enzymes have the same optimal pH and temperature. And why is that? Let's look at our temperature graph first. Let's make sure you understand this. So if we look at different temperatures, the temperatures affect the rate of reaction. So this is how quickly the reaction is proceeding. You could measure that by the change in the amount of product over time, or you could measure this as the amount of substrate used up over time. Either way, you're looking at how quickly is this reaction proceeding. Most reactions are going to have an optimal temperature, and we can see that on the graph as the highest rate of reaction. So in this case, the optimal temperature for this enzyme is really close to 37 degrees. And the importance of 37 degrees is that's our body temperature. So this relates to that question in the bottom right of why might different enzymes have different optimal temperatures? And that's because they're going to match their natural conditions. So over evolutionary time, enzymes are going to evolve to become most functional, most effective at the temperature and at pH conditions where they are found. So enzymes in your body are going to tend to work best at around 37 degrees, whereas enzymes in like root vegetables, for example, in potatoes or turnips, are going to have an optimal temperature that's quite a bit lower. So why does it become less effective at other temperatures? At colder temperatures, it's going to be less effective because all the molecules are just moving more slowly. So you're not going to have things happen as often, and reactions are not going to be taking place as quickly. The reason that the enzymes become less effective at high temperatures is that the enzyme can start to denature at high temperatures. So those bonds holding the protein in the right configuration will start being disrupted. This is the same thing for pH. If we look in that upper right, a change in pH can disrupt those hydrogen bonds, which can disrupt protein folding. So that's true of the hydrogen bonding that's taking place as secondary protein folding in the backbone of the protein, that backbone of the amino acids. But it can also be part of looking at the R groups and how those R groups are interacting in tertiary folding, because some of those R groups are connecting with one another through hydrogen bonding as well. Similarly, quaternary folding, which is, remember, bringing multiple polypeptide chains together, can also be affected because there are hydrogen bonds that are connecting those various polypeptide chains as well. All right, that last graph on the bottom right is looking at substrate concentration. So as we move to the right of the graph, we have more and more substrate. At some point, the enzyme is just going to be overwhelmed, that it can't work any more efficiently if you give it more substrate. So you can think about this like the toothpick case lab that you did last year. So if you had just a few toothpicks around and you were breaking those toothpicks and you're measuring how quickly you can break those toothpicks, adding in a few more toothpicks would make the overall reaction rate faster because you just always had a supply of toothpicks handy. The more toothpicks you have, at some point it's not going to matter because you just can't break those toothpicks any faster. So you could bring in dump trucks of toothpicks and it still wouldn't help you break those toothpicks any faster. Same thing with enzymes. At some point, the enzyme just can't work any faster, and so having more substrate is not going to do anything. Moving on to Unit 3.4, I want you to take a look at these diagrams. So here we have two different reactions. I want you to look just from the diagrams. Do you think these reactions are going to be exergonic or endergonic, and why? So that top reaction is likely going to be an endergonic reaction, and that's because it's an anabolic reaction of making two molecules into one. So we're building a larger product. That next one, where it's showing ATP going into ADP and phosphate, is exergonic. And that's because we're breaking things down. This is a catabolic reaction. So now let's do a little thought experiment. Where do endergonic reactions get their energy? And hopefully this was a little bit of a leading question because you can see that there's one reaction that is requiring energy and one reaction that is producing energy, and that's all we're seeing on this picture. So maybe we can put these together. Another clue is that the title of the slide is coupled reactions. So the idea of a coupled reaction is that an exergonic reaction is going to be able to provide the energy for an endergonic reaction. We can look at that mathematically in terms of the amount of net energy that they either release or require. So here, delta G of the synthesis of these molecules is 3.4 kilocalories per mole, whereas the amount of energy produced by the breakdown of ATP is 7.3 kilocalories per mole. And that's a negative value. If you remember that a negative change in delta G represents an exergonic reaction. So those two reactions, one can power the other. The breakdown of ATP, the energy that is released from that, can power the synthesis of other reactions that are endergonic. So this is called a coupled reaction because they go together. And this is very common in the biological world where you have one reaction powering another. We're going to see that over and over and over. So that's where we're going to leave it off for today. When we come back, we're going to learn about redox, and then we will go into photosynthesis and cellular respiration.